Okay, so this is a video for Module 7, Biology, which is on cellular reproduction and DNA. And if you remember when we talked about the criteria that are required for something to be considered alive, we said that one of those criteria was that the organism needed to be able to reproduce. And you may also remember that we said that reproduction doesn't have to be like reproduction of the whole organism, but the cells, individual cells, need to be able to reproduce. So what Module 7 deals with is the process involved in cellular reproduction, cells being able to reproduce themselves, duplicate themselves so that you end up with multiple copies of the same cell. So at the end of last semester, we finished up by talking about DNA. And you'll remember that that stands for deoxyribonucleic acid. And it is in the DNA that all of the information gets stored to determine different traits and different characteristics. And we also said that basically what DNA does is that it gives the cells instructions for what proteins to make. And it's really the proteins that are made that then determine what function that those particular cells will play. So your hair color is determined by what proteins are produced in the cells of your hair follicles. DNA determines your eye color by signaling that certain proteins are going to be made. So just to emphasize that what DNA does is that it gives the cells the instructions telling the cell what proteins to make, and then it is those proteins that determine what a particular cell would do, whether it be an eye cell or a kidney cell, or determining your eye color or your hair color. All of that's determined by the type of proteins that are made. For many characteristics that an organism has, DNA just determines a general range that you will have of that trait, and then that trait can be, or that organism can undergo some training that might be able to allow it to reach its maximum potential. But DNA sets some limits even on how much that trait can be developed. So for example, if we think about distance running versus sprinting. So somebody who is going to be able to run a marathon, which is 26.2 miles, versus somebody who's going to be able to run short distances like 100 meters very quickly. Well, your DNA will set a certain range. So if your DNA sets you within an average range of being a sprinter, if you train, if you do proper training, you might be able to improve your sprinting ability. So you might be able to get faster, but you'll probably never become an Olympic sprinter if your DNA has determined that you're just an average sprinter. But and the same thing is true that if your DNA determines that you are an average distance runner, you can train and run long distances, and you can improve your ability. You can improve the times that you might be able to run 5 miles or 10 miles or 20 miles. But again, you'll probably never be an Olympic level distance runner if your DNA has not set, because your DNA will set the limits that you can run within the normal range. So for some activities and behaviors, DNA determines your general tendencies, and then your activity or your behavior can determine what part of that set range you end up in. And then there are other traits that are completely determined by your DNA. So for example, the type of blood that you have is determined completely by your DNA. You can't change that, no matter what behaviors or activities you engage in. When we get to the chapter on genetics, which is the next chapter, we'll talk about a certain chemical called PTC that some people can taste and some people cannot taste, and that's determined completely by your DNA. That can't be changed. Whether or not you can roll your tongue is a trait that is determined entirely by your DNA, and no behavior or activities are going to change that. So basically what we are talking about with regard to genetic traits is something that's called nature versus nurture. So nature means that part of traits that are determined by your DNA, and nurture deals more with the environmental factors, like what are your surroundings, what are your experiences, what family activities have you participated in, what choices have you made. So for certain traits, nature sets a general range, but then we can influence that, where we end up in that range, by the particular activities that we participate in. So then when we look if we get back to talking about DNA, you probably have heard the word gene. Well, a gene is just a section of DNA that tells a certain protein or proteins to be produced, and that therefore means that it causes a certain trait. So a gene is a section of DNA that tells certain proteins to be produced, and then those proteins will then determine the trait that the organism would possess. So in discussing reproduction of cells, we're really talking about protein synthesis. And synthesis just means making proteins. Okay, and there are 2 steps that are involved in protein synthesis. Part 1 is called transcription. transcription, and part 2 is called translation. So we'll be looking at each of these steps in turn, keeping in mind that our goal is to take the instructions from DNA to make proteins. So specific cells have specific tasks that they have to perform in order for the organism to survive. So, for example, your skin cells have certain jobs that they need to perform in order for your skin to function and to serve as a protection for your body. And what task that cell performs is dependent on the proteins that that cell makes. So that's why we will look at the steps involved in transcription and translation of protein. In addition to DNA that we've already studied, there is also a structure that is called RNA, and RNA just stands for ribonucleic acid. So here we get the R, the N, and the A. And just like DNA, RNA is a long strand. But if you remember that DNA existed as two strands that form a double helix. So this is DNA, the two strands form a double helix. RNA is usually just a single strand. So RNA is a single strand. Unlike DNA, which exists as a double helix, two strands linked together with each other. Let's review remember we said that the way that DNA, the way that the strands hook up with each other were by their bases. So for DNA, we learned that adenine links with thymine. And we learned that guanine links with cytosine. And by these pairs of bases linking up, then DNA forms its double strand. For RNA, RNA also contains those nucleotide bases, but they are slightly different because RNA, so the bases for RNA are slightly different. We still have adenine. We still have cytosine. And we still have guanine. Okay, so those first three bases are the same ones that are present in DNA, but in RNA we have the fourth one is different, and it is called uracil. So there's no thymine in RNA. Instead, the thymine is replaced by uracil. So now we need to ask ourselves, why is RNA important in making proteins? So what is the role of RNA in making proteins? So remember that DNA is DNA is in the nucleus of the cell. And yet we know that proteins are made in the ribosome. So if DNA's job is to give instructions for what proteins to make, and DNA is in the nucleus, but proteins are made in the ribosomes, there has to be some way to get the information from the nucleus into the ribosomes, which is outside the nucleus, obviously, so that proteins can be made. So to get the information from the DNA in the nucleus to the ribosomes, there is a type of RNA that takes a picture of the DNA and then carries that information to the ribosome. So RNA takes a picture of the DNA that is present in the nucleus, then the RNA leaves the nucleus and it carries that picture of the DNA to the ribosome so that the protein can be made. And the way that it takes a picture is the same process that we've seen before. It takes a picture by linking bases together. So if we have a strand of DNA in the nucleus, that looks like this, guanine. So we want to take a picture of this strand of DNA. If it's guanine, thymine, and adenine, then RNA would link up with this strand of DNA by linking its bases. So we know that guanine always links with cytosine. And we know that the thymine that is in DNA will link with adenine. And now this next one's a little bit tricky because we said that RNA doesn't have thymine, it has uracil. So if there is an adenine on the DNA strand that needs to be copied, then the base that links with that for RNA is uracil. Okay, so it's not going to link with thymine because there is no thymine in RNA. Okay, so this is the DNA strand that we wanted to copy, and the RNA copies it by linking with the corresponding bases that would link together. So let me draw a strand of DNA that would show its bases, and then we'll look at the RNA bases that would link to it. Okay, so let's say we have, here's a strand of, let me erase that. Oh. Okay, so here's a strand of DNA. Okay, so this is all DNA. So let's say we have, this is guanine. So for DNA, guanine links with cytosine. Thymine links with adenine. Adenine links with thymine. Cytosine links with guanine. Okay, and then when we have a strand of DNA and we want to copy a certain portion of that DNA, the first thing that happens is the portion that's going to be copied unravels. So that's what I've shown here on the bottom, is that the portions of the DNA strand that we want to copy unravels. So if this DNA strand is cytosine, adenine, guanine, thymine, and then we'll put one more in there, we'll put an adenine. And then this portion of the DNA over here would have the bases that correspond to that, but we're not concerned about that right now because this bottom portion here on the left is the DNA strand that we want to copy. So what will happen is that you'll have an RNA strand that will come in and it will pair with this section of DNA here that we want to copy by linking with its corresponding bases. So the RNA strand, we said that cytosine links with guanine. Adenine is going to link with uracil because there's no thymine in RNA. Guanine will link with cytosine. Thymine links with adenine. And then again, this adenine will link with uracil because there is no thymine in RNA. So this is the RNA strand that has been copied from the DNA strand. And this RNA strand will then be carried to the ribosome, then travel to the ribosome so that the specific protein can be made. And that process that I just showed you is the first. Remember I said that there's two steps. The first step is called transcription. And so that process that I just showed you is the process that's called transcription. So it's the RNA strand linking up with the portion of the DNA strand that needs to be copied so that then the information can be carried to the ribosome for protein to be made. Okay, so if you have let's just run through one. If you have a DNA strand that is comprised of adenine, thymine, cytosine, and another adenine. Okay, so this is the DNA strand. What would the RNA strand look like? Well, we know that adenine for the RNA is going to link with uracil. Thymine will link with adenine on the RNA. Cytosine will link with guanine. And this last adenine, again, there's no thymine on RNA, so that would link with uracil. So this then would be the strand of RNA that would be made in order to duplicate this strand of the DNA. So next I would like to show you a figure. Now, this is just a figure, and this is from your textbook if you want to refer to it. So this is on the left side is a DNA strand, and you can see that the DNA strand it forms a double helix because the bases join together here. So these bases you can see all would link up together. But we want to duplicate this section of the DNA. So that section of the DNA strand is going to unravel, and we've seen that happen here. So now if we look on the right side, then the RNA strand will come in and it will pair with the bases to essentially take a picture of this DNA strand. So cytosine joins with guanine, adenine joins with uracil, guanine, cytosine, guanine, thymine on the DNA will bind with adenine on the RNA, adenine will bind with uracil, and etc. So this DNA strand has now taken a picture of the DNA strand that is to be copied so that it can then carry it to the ribosome so that protein synthesis can take place. So the other thing I want you to notice is, so this is inside the nucleus here, but the thing I want you to notice is that the membrane that goes around the nucleus, so this is the membrane that goes around the nucleus, and what you should notice is that there are holes or openings in the membrane that goes around the nucleus, and those are called pores. So there are pores in the nuclear membrane, so that now that we've taken a picture of this RNA strand, that RNA strand can leave the nucleus by going through one of these pores. So that is that's a very important part of the DNA synthesis, is being able to remove that, get that RNA strand out of the nucleus so that it can then go to the ribosome. So here's that same figure again. You can see the RNA strand duplicated, and then that RNA strand would be able to leave the nucleus through one of these pores. Okay, so then the next process that is involved in this figure, and now we know that the next process that is involved, so that was transcription, and now the next step is called translation. And just like when transcription takes place in the nucleus, translation is going to take place in the ribosome. And the piece of the portion of the ribosome that left the nucleus and goes to the ribosome is called messenger RNA. So because it's carrying the message from the nucleus to the ribosome so that the protein can be made. So translation, when we do translation, then there is going to be another section, another portion of the RNA, and that portion of the RNA is going to be called transfer RNA. So in the ribosome, the ribosome is going to have three things. It's going to have amino acids. And remember that all a protein is different amino acids linked together. And the types of the particular amino acids that are linked together and how many of them there are determine what the protein is. And then there will be enzymes. And remember previously we said that enzymes mainly serve as catalysts, so they speed up chemical reactions without being changed in the process. And then the other thing that will be in the ribosome will be the transfer RNA. And just like the messenger RNA, the transfer RNA will also be made up of those bases. But each transfer RNA is also bound to an amino acid. And this will make sense in just a moment when we look at a figure. So in the ribosome, you'll have these amino acids, and the amino acids are going to be bound to the transfer RNA, and then you're going to have enzymes that will just help just speed up the process so that protein synthesis can occur faster than it would under circumstances where those enzymes were not present. Okay, so this is a figure that is going to show how the bases on the messenger RNA and the transfer RNA link up in order to produce proteins. So on the top of this figure is the messenger RNA that came from the nucleus. So we can see here, so here's adenine, uracil, guanine, cytosine, guanosine, guanine, guanine, cytosine, uracil, can't see what that one is, it might be uracil, guanine, cytosine, uracil, uracil, adenine, uracil. So this is the picture that was taken of the DNA by the messenger RNA, and now it's been carried into the ribosome. When this messenger RNA gets to the ribosome, it will link up bases with the transfer RNA. So this RNA that is being shown here is the transfer RNA. And you should be able to see that so in this particular case here, the cytosine attached to guanine, guanine with cytosine, cytosine with guanine with cytosine. And then over here, cytosine and guanine, uracil, adenine, uracil, adenine. So if we just pay attention to this particular section, you can see that the appropriate bases lined up with each other. And the other important thing to notice is that for every 3 bases on this transfer RNA, that base carries with it an amino acid. So in this case here, the cytosine, guanine, adenine section of this transfer RNA carries this amino acid alanine with it. So when this section of messenger RNA binds with this section of transfer RNA, it's going to bring with it this amino acid arginine. Then when this section of messenger RNA pairs with this section of transfer RNA, it's also going to carry an amino acid with it, and that amino acid is leucine. So now just with these 2 sections pairing up, we have 2 amino acids that have lined up as well. And these 2 amino acids, if that's all it is, they make up a certain protein that would then perform a certain function for the organism. If we did this whole section of messenger RNA, then for every 3 bases, you would have another amino acid that would be carried along with it. So in this 5, there are 5 groups of 3. So with this section of messenger RNA, we would get 5 amino acids lined up, and the length of the messenger RNA would then determine how large the protein is that is made and what particular amino acids are linked up together. So this figure that I am showing now then shows the whole process. So in this section right here, we have the DNA strand that we want to copy. So we're interested in copying this strand right here. So the first thing that happens is the DNA strand unravels. Then messenger RNA comes in and it takes a picture of that DNA strand by linking its corresponding bases. Then this figure then shows that strand of messenger RNA leaving the nucleus through a pore, and they will then travel to the ribosome. And then in the ribosome, this is our strand of messenger RNA that came from the nucleus. It will line up with the corresponding bases, and for each group of 3 bases, there will be 1 amino acid attached, and they're showing that here. So that with these 3 bases and these 3 bases, each one's going to have an amino acid attached to it. So again, depending on how many groups of 3, you're just going to have a long strand or a short strand of amino acids linked together, and that is what a protein is. So when all of the amino acids that are called for are linked together, then that gives you a protein, and that process is called translation. So just to review, transcription Transcription is the process of the messenger RNA making the copy. So messenger RNA copying the DNA in the nucleus, and then translation is the messenger RNA leaving the nucleus going to the ribosome lining up with the transfer RNA. And remember, the transfer RNA has the amino acids linked to it. So then you get the amino acids linked together. And that is what gives you the protein. And the protein that is made then determines what particular function that type of cell will perform. Okay, I think we're going to stop there for today, and the next time we will look at the process of mitosis, which is the process that is involved in reproducing, and in this case, reproducing eukaryotic cells. So we'll stop there for today.